Hello, welcome. I'm Bellington Kamisa. This is biology form 3. I'll take you through transport in plants, but basically looking at a subtopic which is osmosis and active transport. As you can see from the pictures, it shows that it involves movement of water entering the roots and being transported to somewhere there. We are on the success criteria, the objectives that you are supposed to achieve in this lesson. So we are saying by the end of this lesson, you must be able to define the term osmosis. You are supposed to state the importance of osmosis. You have to conduct an experiment on osmosis in living tissues using potatoes or purple petioles. You have to explain active transport in plants. You have to state the importance of active transport. Here we are on osmosis. We want to carry out an activity below to learn about osmosis. First of all, this activity goes like this. You have to demonstrate osmosis using the Visking tubing. What materials would you require? For example, these materials that one would require are Visking tubing, 1, 2, concentrated sucrose strokes. You can also use sugar solution. Then in number 3, beaker filled with distilled water and the string. Right, the procedure now, the steps that are followed are as follows. Number 1, you measure and cut an 8 centimeter length of Visking tubing. Number 2, you tie one side of the Visking tubing tightly with a string one end from the end. So you fill the Visking tubing about 3/4 full with the concentrated sugar solution. Then you tie the other end of the tubing tightly with a string. Number 5, you have to immerse the Visking tubing with the sugar solution into a beaker full of distilled water. Then you note the level of water in the beaker. Then you have to leave the setup to stand for several hours. Then you note the amount of solution in the Visking tubing and the level of water in the beaker. Now let us see what could be the results. Now here on these slides, we are looking at the results of osmosis from that experimental procedure that we have elaborated, but now we are going to see what would be the effect. Now here there is glucose inside the Visking tubing, so this is a Visking tubing, and indeed this one is a beaker. The whole of this beaker is filled with water, and indeed this is water, but only here we have the Visking tubing. So the Visking tubing is acting as a semi-permeable membrane which can allow some molecules to enter the Visking tubing and others not allowed. This water is distilled, that's the pure water, and here the same, this is the beaker, but in this case we have the Visking tubing which has bulged. So you can see that something has happened here. Water might have entered. There is sucrose here, which means there is low water concentration in the Visking. So therefore, water enters the Visking and now it budges. It becomes, it is expanding, but the best way we say it budges, it swells up. So you can see that the Visking tubing now swelling up. Questions that guide the activity. What happens after several hours? So you have to suggest answers. What has happened? Contents of the Visking tubing, water in the beaker, and again you have to repeat the procedure 1, but this time you fill the Visking tubing with distilled water, then tie tightly until it is uh, uh, until it is now fixed and budging. Then you immerse the whisking tubing into a beaker of concentrated sugar solution and leave it to stand for several hours. Now the results would be, before the experiment, you can see that now here we have this, the whisking tubing, for example, is placed here. So inside here now we are putting the water here, then we have the strong sucrose solution inside, so water enters. So that's why the level of water must drop in here, because the whisking tubing gains water by the process of osmosis. Questions: what happens to the contents of the whisking tubing after several hours? Does it still feel firm to touch? Does it still appear budging? Explain your observations. Now, in the activity of demonstrating osmosis, water also moves from a region where it is in high concentration to a region where it is in low concentration. So you can see inside the whisking tubing, we have sucrose or sugar solution. So therefore, we have less water molecules. That's why water moves from the beaker into the whisking tube through this impermeable membrane. So therefore, osmosis is also a type of diffusion. However, the term osmosis is used only when we are referring to diffusion of water molecules through a semipermeable membrane. So therefore, we can say osmosis is diffusion of water through a semipermeable membrane to other side or into the cell. So in our experiment, the whisking tubing act as a semi-permeable membrane. Now, osmosis is the movement of water molecules from a region of high concentration to a region of low concentration through a semi-permeable membrane. So it means water moves into the cell and out of the cell through the membrane. And this membrane is impermeable, meaning to say it is selective, can allow some small molecules to pass through, and those that are large are not allowed to pass through. So water molecules are at their high concentration in dilute solutions. So you can see, if you have diluted, for example, sobo in our homes, if you have sobo, that sobo, before you add water, is having more solute dissolved in that. So if you add more water, you dilute it, it means you have therefore more water molecules. So water molecules are at their high concentration in dilute solutions. That's what we said. So these are solutions with more water and fewer dissolved solute molecules, e.g. salt solution. Right? Water molecules are at their low concentration in concentrated solutions. Now these are solutions with less molecules, e.g. solute molecules. The whisking tubing separates such two solutions. Water moves from the dilute solution to the concentrated solution through the small pores of the semipermeable membrane. So a semipermeable membrane is a membrane with very small pores. So being very small, it means the large particles cannot pass through them. That's why they are selective. So these pores allow only those substances with small molecules to pass through. So substances with large molecules like the solutes do not pass through these small pores. So as you learned earlier on, cell membrane is impermeable while the cell wall is permeable. So the cell wall, it means is porous, can allow even some large molecules to pass through. So permeable means that it has large pores that allow substances with all sizes to pass through. So the following diagram illustrates the process of osmosis. Now, the figure below illustrates osmosis through semi-permeable membrane. So this one is acting as a semi-permeable membrane. And here we have the sugar solution. So the sugar solution is having the large particles. These particles are for sugar solution. And the small particles, these ones are for water solution. So here, this side, we have distilled water. So therefore, this distilled water moves through these small tiny pores to this other side. So this movement of water molecules is called osmosis. And however, some water molecules may misbehave. They may move from this sugar solution going that side. So that's why at times osmosis is defined as a net movement of water through a semi-permeable membrane from a region of high water concentration to a region of low water concentration and vice versa. Yeah, so that's it. Now we have to conduct an experiment to show that osmosis is taking place in actually living tissues. For example, one would need the following materials: fresh cassava, can use sweet potato, Irish potatoes, can use salt, strong salt solution, can also use distilled water, scalpel. A scalpel is a cutting material in the laboratory. So we have also large beaker or a small basin. These are the materials that one can use to demonstrate that osmosis is really taking place in living tissues. The procedure. What steps are taken to do that? Number one, you take a large Irish potato and peel it. Number two, you could also use cassava or sweet potato. Alternatively, you may use one here, a sweet potato or Irish potato or cassava. Each one of them will work. Now, you cut off a piece so that it stands at least a height of 6 centimeters. You cut and scoop out a deep hollow portion in its middle and pour the strong salt solution halfway up the hollow portion. Now you mark the level of the salt solution using a scalpel. That's number 3. Now number 4, you place the potato in a beaker or basin containing distilled water and let it stand for several hours, then note the level of solution in potato. Then number 5, you repeat the experiment with boiled pieces of Irish potato or cassava. Now here is the experimental setup. So it shows here we have strong salt solution. We have the living potatoes, this and that living potato, and this one is boiled potatoes, and you have distilled water in the beaker. This is distilled water, but mind you, the boiling kills the cells, and therefore, because the cells are killed, we expect no movement of water in those cells. They cannot take up water. Right, here we have, for example, we have strong salt solution, which is there. Now, this salt solution now will go this way, where we have now the solution will rise here. All these grain-like here, now they are dissolved. So you can see we'll have the collection of the liquid will rise here. And there will be a drop in distilled water in this particular beaker A. Here nothing was put and there is nothing collected. Here strong salt solution, and here the result there we have the same, nothing has changed. So because this potato was boiled. Now let us look at the question: is the level of the strong salt solution still the same at the end of the experiment? You can guess. What are the answers for this? You can try to guess before you go to the next slide. Explain what happens to cause the change in level of the salt solution. You compare these results with those obtained when boiled Irish potato is used. Now, if a concentrated solution is separated by a semi-permeable membrane from distilled water, a force pressure develops. Now, the force draws water molecules from the distilled water until an equally concentrated solution is attained, which is isotonic solution. So the pressure is known as osmotic pressure. Now, this shows that a concentrated solution has hidden pressure which draws water molecules while separated by a semi-permeable membrane from a less concentrated solution. This hidden pressure is known as osmotic pressure. So it's osmotic pressure, we are saying it's a hidden pressure that is exerted by the highly concentrated solution through the semi-permeable membrane. Now let us look at there are types of solutions that can be separated by the membrane. We have what we call an isotonic solution. So isotonic solution refers to two solutions which have achieved the same concentration. So isotonic concentration, we mean the same concentration. Now the two solutions have the same osmotic what? Pressure. So it means there is a balance in pressure. So therefore there is no movement of water. So we have another type of solution, which is B. Isotonic is A, and B, we have hypotonic solution. So this one is a lowly concentrated solution, a very lowly concentrated, means it's a low concentration. Hypo, the prefix hypo, it means low concentration. So therefore, when a cell is placed in a such solution, gains water by osmosis. Now, a hypotonic solution has low osmotic pressure generally, so therefore the low concentrated solution usually when a cell is placed there, it usually gains water. So a hypertonic solution is a solution of high concentration. So a cell placed in such a solution loses water by osmosis. Loses water by osmosis. So a hypertonic solution has high osmotic pressure. We have also an activity which can help us to demonstrate osmosis in petioles. For example, the one would need the following materials. One would need leaf petioles from purpose, salt solution, distilled water, petri dishes, labels, then the scalpel. The procedure, the steps that are followed to demonstrate osmosis in living tissues such as the leaf petioles. Now, number one, you cut petioles from the three leaves from purpose, for example. Now, number two, you split each petiole lengthwise, you cut along and longitudinally, and you place the two strips from the same leaf in water, salt solution, and in another one, an empty petri dish in that order. That's to say, respectively. So you leave them for 30 minutes. Then number four, you draw the appearance of the petioles after 30 minutes in each solution. So you have to draw the appearance. How do they look like? So we can see theoretically how these drawings respond, or how these diagrams will appear at the end of the day. Now, questions that will guide your understanding. Number one, you explain the appearance of each petiole in the solution. It was mixed. So number two, you explain why it was necessary to split petioles in this experiment. Those are the questions. Can you guess the answers for these to understand the content? Now let us look at the explanation that is based on what we have done under the procedure. Now the first one is the inner cut surface of the strip of petiole is described as the inner side. its cells are exposed because you had to cut them lengthwise. So the outer surface of the petiole is covered by a thick cuticle. So when a strip of petiole is immersed in water, the cells in the inner side exposed cells take up water by osmosis much faster than the cells on the outer covered by cuticle surface. So these cells expand more, causing a curvature, a bend towards the outside, that's to say outwardly. So the cells expand and then they bend outwardly. So when the strip is immersed in salt solution, the cells on the inner side lose water much faster than the outside by osmosis. So the cell in the inner side shrink more, they reduce in size, and hence the strip of petioles curves inwardly. So the strips in the empty petri dish do not change because there is nothing, they are neither losing water nor gaining water. Usually this is used for comparison purposes and is referred to as the control experiment. So the strips that are put in the empty petri dish, they are acting as a control experiment. So the control experiment generally is used for easy comparison so that you can compare the results. Significance of osmosis. Why is osmosis very, very important, very vital? One, osmosis is involved in absorption of water by plants in the roots. One. So it facilitates the uptake of water by the plant roots. Number two, it is also involved in turgidity. When the cells have gained water, they become turgid. The cells expand, they branch, they swell up in the long run, supporting the plants. So plant cells obtain water by osmosis to become turgid. That's the biological term that we use. So when they are touched, when the cells are touched, they become film, providing support to the plant tissues. So guard cells obtain water and also lose water by osmosis to enhance opening and closing of stomates. So generally, guard cells, when they lose water, the stomates close. So that's one of the importance of osmosis. It facilitates the closing and opening of the stomach. Now let us look at active transport. This is another physiological process, but the type of movement of molecules and ions that we have considered so far are those where molecules move down or along the concentration gradient. But now, these movements are called passive transport. So we have looked at diffusion. That diffusion is an example of passive transport. We have also looked at osmosis, which is an example of passive transport. We have systemic streaming, cytoplasmic streaming. Molecules and ions can also move from an area of low concentration to that of higher concentration. So they are said to move against concentration gradient. Such a process requires the use of energy and is called active transport. So we can see that now active transport, the ions and molecules, they move against concentration gradient, which requires a lot of energy. Expenditure of energy is required to transport the needed materials. Now, in active transport, the cell must use its own energy to move the molecules against a concentration gradient. So such energy is supplied by the mitochondria in the cell. Therefore, active transport takes place only in living tissues. So mainly, active transport takes place in living tissues. In living cells, once the cells are dead, generally active transport may not take place. The figure here just illustrates or shows osmosis and as well as active transport taking place. So you can see this figure here shows a semi-permeable membrane. So here, this side, we shows water molecules, and they are moving through this semi-permeable membrane to this side here. So now this side, we have active transport taking place. Now the ions or the molecules are passing through this to where there are already more. So here you can see we have only 3 ions here, or molecules, that are being taken to where there are already more. So they have to move by active transport. So you can see this diagram indeed. So the diagrams have been adapted from book 3, Excel and Succeed, page 73. So you can also refer to those diagrams with other diagrams. So active transport involves carrier protein molecules. They pick up molecules of such substance from one side of the cell membrane and transport them across. Significance of active transport. Why active transport? Very important. Number 1, active uptake of minerals. So active transport facilitates the absorption of mineral salts from the soil into the root hairs. So as mentioned earlier on, the concentration of salts in the cell sap of the root hair cell is higher than that of the soil water. So soil water is dilute solution of mineral ions such as potassium, magnesium, nitrates, and phosphates. So once there is basically we have dilute of those, it means they are less in the soil. But in the cell sap of root hairs, we have plenty of them. So therefore, those ions still they are needed by the plants. They move by the process of active transport. And this one requires expenditure of energy, which is provided by mitochondria in cells. Yeah, that's it. Let us go to the next slide. Here now we have this. This means that a root hair must take up, the root hairs must take up these mineral ions by active transport against their concentration gradients. Now, plants need mineral salts for normal growth and development. Remember, in the previous lesson, we mentioned of nitrogen, for example. Nitrogen, in form of nitrates, is combining with glucose to produce protein for growth and development. So, for example, they need magnesium to synthesize chlorophyll. And chlorophyll is a green pigment which is needed to trap sunlight, which is a necessary condition for food making process. So, and the nitrate ions to manufacture, to synthesize means to manufacture proteins. So you can see, I already said that the nitrates combines with the glucose to produce proteins, which is there to promote growth and development in plants. Number 2, the second function of active transport is transport action of manufactured food. Remember, the glucose is made in leaves and it has to be transported from the phloem into the sieve tubes. So in sieve tubes we have abundant, and this requires active transport. So we are saying translocation is the transport of organic substances. Basically, these organic substances are food substances from one part of the plant to another. So it takes place in the phloem tissue in plants. So the substances are transported to tissues where they are required for body processes in the plant. For example, respiration. So the glucose is required for the process of respiration to generate energy, which is very much needed in active transport and other processes. Now, number 3, the significance. Remember, we are looking at significance of active transport, and the third importance is that it facilitates the storage of substances in tissues. So we are saying Active transport is involved in transport and accumulation of substances in storage tissues. What are these storage tissues in plants? You can guess, I think. So you have some of tubers, seeds, as well as fruits. We consume fruits. Don't you consume yourself? You can see you consume fruits. Now, with me, I have the practice exercise that will cement your understanding. Make sure that you attempt these questions and you provide feedback so that we compare with our own responses. Number one, you define the following terms: A, diffusion; B, osmosis; C, active transport. Number two, you have to state two significances of A, diffusion; B, active transport. Number three, what factors influence diffusion? Number four, what is semi-permeable membrane? Number five, you have to describe an experiment that you can conduct to show that osmosis really takes place in living tissues. You have to write your answer in essay form, and this response should include the materials, procedure, results, and conclusion. For resources, we have references. Now the books that has been used, we have Cerebral 2017 Excel and Succeed Biology Book 3. We have also used Chimocha Similon 2017 Achievers in Biology Book 3. For illustrations, design, and graphics, these have been done by Charlotta Bruni and Dave Mumba on work on Xulendo. Then we have also photos have been taken on this internet. We have this and that one. These are just links where we have extracted some diagrams, pictures. Then we have Excel and Succeed Biology, page 71. You can check from your diagrams. That's where we have taken the diagrams. These are pages for the diagrams where you can access these diagrams.